Good morning, everyone. Welcome to another lecture in the series of molecular biology. See, in past few lectures, what we have done actually, we have established the complete understanding of basic pathways like DNA replication and other processes like transcription, post-transcription modifications. We had also discussed translation process, but we had discussed it in terms of prokaryotes. So today we will be focusing on translation process in eukaryotes. See, there is no much difference when we are going for the comparison between prokaryotes and eukaryotes. But yes, in terms of the initiation of translation, there is a huge difference. There are multiple players involved. In prokaryotes, we saw that there are only three initiation factors. In the last lecture, we had narrated that we had established that complete understanding that yes, there are only three initiation factors that are involved in initiation of translation or initiation of protein synthesis in prokaryotes. But when you're talking about eukaryotes, there are multiple players. There are nine initiation factors that are involved, each performing specific function, and with the main aim is make a pre-initiation complex followed by a initiation complex, a translation initiation complex that is called as ATS ribosomal initiation complex. So, what is our motto in this particular lecture? What is our aim? Our aim is to have a clear-cut understanding that how the translation process is happening inside eukaryotes, what similarity it is having with the prokaryotes, and what are the dissimilar features, what are the distinguishable features, what are the roles of initiation factors and the other factors in the later stages of elongation and termination. And then we will be going a step ahead in just having a brief that once a peptide chain is formed, what is the fate of that peptide, how it is folding, what are the post translational modifications that we are talking about. So by the end of this lecture, you will be able to know all these phenomena in a very depth and detail, and the complete chronology of the events that is happening happening in this particular process of protein synthesis. I hope that will be clearly narrated by the end of this lecture, and you will be having a better understanding of that topic. So when we are talking about the translation in eukaryotes, see, first of all, it should be very clear in mind when we had discussed prokaryotic translation, we had clearly established that the first amino acid residue that starts this particular process is formilated methionine, and there is a specific tRNA molecule which recognizes this formilated methionine, which after joining is called as formilated methionine tRNA. In eukaryotes, there is no formilated methionine when we are talking about the translation of the nuclear genome or the mRNA which is made from nuclear genome. So when we are talking about the nuclear genome genetic information transformation, the first codon always codes for methionine, but that methionine is never formilated. That is the first distinguishable feature when we are talking about in terms of prokaryote to eukaryote. Yes, the first codon will always be AUG. It will be coding for methionine, but it will be never coding for any formilated methionine. But then again, our eukaryotic cells, like plant cells have chloroplast and mitochondria, and the animal cells that we are talking about in terms of mitochondria, these organelles have extra chromosomal DNA, right? And that extra chromosomal DNA also goes for transcription by forming mRNA, and then it goes for protein synthesis by a translation process. So if you observe that, but the translation process of these two organelles, you will see that the first codon, that is AUG, which is present in the mRNA, that codes for formilated methionine. That means the organelles which are present inside the cell, which are carrying the DNA, that is chloroplast and uh, mitochondria, when they are forming the proteins, the first amino acid is always formilated methionine, and that is another proof which augments that theory that the mitochondria and the chloroplast are from prokaryotic origin, or bacterial, or the bacterial ancestors shares the same process that is happening in prokaryotes. That's why people have advocated that mitochondria and chloroplast originated from bacterial ancestors. So that is the reason that in eukaryotic cell, when we are talking about the translation process, it is going as a prokaryotic in a prokaryotic manner, similar to prokaryotic manner in terms of organelles, and quite similar to the prokaryotes in nuclear genome also. So let us go ahead and have an overview. Again, the translation process is divided into three stages. We will divide in the initiation, elongation, and the termination. And then all the stages, see all the stages, what is the prerequisite requirement? As we had discussed in in the case of prokaryotes also, we need mRNA, we need ribosome, we need tRNA, and we need amino acids. That is the basic requisite. Apart from this, what is required? We require multiple initiation factors, and the ribosome, which was obviously present in the prokaryote, was of 70s, but in eukaryotes we'll be talking about 80s, and that 80s is present in form of 60s and 40s. Now, in eukaryotes, these ribosomes are not in associated form. These are present as a separate soluble subunits as 40s and 60s. They will come together only in the case of ribosome to give rise to a only in the case of translation to give rise to a compact ribosome, that is 80s ribosome, eukaryotic ribosome. So this assembly of 40 and 60 to give rise to 80s requires all these molecules, the initiation factors, the mRNA, the tRNA. Then only this 80s ribosome unit is formed. This 80s ribosome unit is formed. So when you'll see that how this process is starting, you will be able to know that first of all the small subunit will be coming, and then the mRNA and the charged methynyl tRNA will be coming. They will be forming a complex, and to that complex the larger subunit is coming later on and binding to give rise to a translation initiation complex. And that is the main motto. That is the main motto, to understand that what is happening in the initiation process. Now, another peculiar thing that one should be able to clearly understand before we dive directly into the translation process. And what is that thing? What is that thing? The thing is very clear. See, in prokaryotes, I told you in earlier lectures also that in prokaryotes, the translation is happening simultaneously with transcription. That means as soon as the mRNA is formed via transcription by RNA polymerase, immediately the 5' end is attacked by the ribosome. The 5' end is attached to the ribosome, and the protein synthesis starts, the polypeptide chain synthesis starts. Whereas in eukaryotes, first the mRNA is released, then it is modified, and then after modifications, post-transcription modification, it is released in the cytoplasm, where it will go and bind to the ribosome to give rise to protein synthesis. So the peculiar thing that is there in eukaryotes is that because of the presence of initiation factor, the linear mRNA which is formed takes a transient loop structure, a transient circular loop structure. And how that circular loop is forming, how that loop structure is forming, if you see, then the elongation, the initiation factor, the EIF represents here the eukaryotic initiation factor. Now the EIF family, or mainly the EIF4 family, the initiation factor 4 family, and in that 4EG, these two proteins play a very crucial role. So what happens? See, at the 5' end, the 5' end of the cap, there is a cap of mRNA. The mRNA at the 5' end has methylguanosine, which is acting as a cap. This cap will be recognized by the initiation factor 4E, whereas the poly-A tail is associated with multiple proteins called as PAB, poly-A binding proteins. So at the terminal, suppose this is the mRNA, at this terminal I have E, at this terminal I have PAB. So if somehow this protein goes and binds to this protein, then it will form a loop. It will form a closed loop, and that is what happening. The IF4G, which is again an initiation factor, it bridges the gap. It binds to 4E at one end, and at the other end it binds to poly-A tail binding protein. Thereby it circularizes the linear mRNA, and that is a transient state which is achieved in initiation complex, and it always happens at 40s ribosome subunit. So we'll see that how these things are coming about. So first of all, it is very important for you to understand this, that the initiation factor 4E and G are performing this factor. But I told you that we have 9 different initiation factors, right? 9 different initiation factors. Then let us see what exactly are we talking about. What are the functions of these of what are the functions of these initiation factors when we are talking about in terms of eukaryotes. See, here I have provided you the list of all the 9 initiation factors, and when we are talking about IF2, that is initiation factor 2, 2b, and 3, these are performing the same functions as was the case in case of prokaryotes with the 1, 2, and 3 molecule. There we observed that one IF1 is binding to A site, the IF3 will be binding to the small ribosomal unit, and IF2 will bring the methonyl formalated methynyl TR into the P site. And here also is the same scenario. What is the scenario? The initiation factor 2 will bind to the 40S ribosomal unit, and this initiation factor 2 is actually the molecule which brings the first minor-cell tRNA, that is methonyl tRNA, and attaches it to the P site, and it is bound with GTP molecule. Then we have IF2B and IF3. These are the factors. In fact, IF2B and IF1 and IF1A is also there, which is nomenclature in that manner. Then these are the initiation factors that will first go and bind to the small ribosome unit. Why they are binding? Because they are blocking the site. They are blocking the non-specific site. They will be blocking the A site. They will be blocking the E site, so that only P, that is peptidyl site, is available for binding to the methonyl tRNA. And IF3 will go and bind to the ribosome, 40S ribosome, so that premature binding of large and small subunits should not happen. Then we have IF4A. Now this IF4A is again a unique initiation factor. It's an enzyme. It's actually a RNA helicase which will open up the double strand RNA. Now you will ask me, where is the double strand RNA? We have only single strand mRNA. Then where is the double strand RNA? See, in the 5' region we have untranslated regions, UTRs, and that we have observed that that particular segment gives rise to a super secondary structure, that gives rise to a secondary structure, a loop kind of structure. And in that certain small portion, the single strand orients itself in such a manner that it forms a very small stretch of double strand RNA, and that double strand RNA is required to be removed and in order to linearize the complete mRNA. And that is the function of this particular initiation factor 4A, which is acting as a helicase. Then we have IF4B, 4E, and 4G. Now this 4B, what it does? Once the ribosome has bound to the mRNA, once this ribosome small subunit has bound to this mRNA, and suppose the starting is to be performed by this particular origin, then what will do? It will bind to the mRNA. Now it has to scan the mRNA in order to reach this start codon from where the protein synthesis will start. Then this scanning of the mRNA is mediated by initiation factor 4B. Then we have 4E and 4G, what we had read in the last slide. This 4E and 4G, see, 4E binds to the cap molecule and 4G bridges the bond between this 4E and PAB protein, so that the complete mRNA is now circularizing. Then we have another initiation factor which is very important in my terms in case of translation in eukaryotes, that is IF5. Now this IF5 goes and binds to the small ribosome unit. Then what it does? It promotes the dissociation. Once there is a signal is given that now the big ribosome unit, that is 60S, will be coming and completing the complex, so that it requires certain place to bind. But we have all 7 initial factors which are bound there, so there is no space. So first, the IF5 is responsible to clear out all these initiation factors and dissociate them from the small subunit, so that the larger subunit can easily come and bind. So that's why it is a very important initiation factor. Then we have initiation factor 6. What it does? It facilitates the dissociation. Once the complete ribosome has done its job, then it is inactivated, or its inactivated form. Then the dissociation of those two subunits is mediated by IO6. So you see, that is the case. But once you have to understand that when we had talked about this particular IF4B, the initiation codon is although AUG, but the mRNA is detected at a different place in close proximity to your Schindel-Garno sequence. But then again, that is not the site, but it is the job of IF4B to assist the scanning of mRNA by small ribosomal unit and reach to the start codon in order to initiate that particular protein synthesis. And once it has aligned to that to the starting codon, that is the signal that yes, now the protein synthesis is going to start. Now the protein synthesis is going to start. Now this is all about, you know, the complete what are the roles that are being played by the initiation factors, and how these initiation factors are bringing about the different activities that are very much required in order to have an efficient translation, now the efficiency, when we are talking about of translation, the efficiency of any translation process depends on multiple properties. It depends on majorly depends on what RNA is, what mRNA is coding, how complex will be the protein that will be formed, what will be the length of the coding protein, and also what is the length of the polyA-tail. In majority of the cases, longer the length of the polyA-tail, better is the efficiency. Then how efficient is your initiation factor for 4A, which is an helicase that will be removed, that will remove the double strand part. So all these factors cumulatively are responsible to execute an efficient translation process in eukaryotes. Now without any delay, after knowing all these functions of initiation factors, now what is our job? Our job is to arrange these functions in a sequential manner, how exactly the way it happens in eukaryotes. So let us start with it. So let us start with the pictorial representation only. We'll be discussing this pictorial representation, and in the subsequent slides we'll be having that particular related text if you want to have any kind of references in that section. So let us discuss this particular section. We have a small ribosomal subunit, right? We have a small ribosomal subunit, that is 40S. Now in eukaryotes, this small ribosomal subunit has three sites: A aminoacyl, P peptidal, and E exit. Whereas in prokaryote, we saw that the small ribosomal subunit, that is 30S, had only two sites, that is A and P, and the E site was brought about by prokaryote, by the larger subunit. But in eukaryotes, the smaller ribosome unit also have all the three sites. That's first thing first. Then the first initiation factors that come to play are 1 and 1A. What it will do, 1 and 1A will come and bind on E and A side respectively. Why? Because I told you that the first amino acyl tRNA, that is methonyl tRNA, will always come and bind to P side, and all the other remaining amino acyl tRNA always come to the A side. So in order to ensure that the binding of the tRNA should happen at P site only, and it should not enter in on E or A side. So what eukaryotes are doing? Eukaryotes are blocking this site. Eukaryotes are blocking the E site and the A site, so that only P site is available. Then there is another initiation factor, that is EIF3, eukaryotic initiation factor 3, which binds to the small ribosomal unit and prevents the premature binding of large and small subunit. Then simultaneously, what is happening? The initiation factor 2, which is a GTP bound molecule. It will bring the first methonyl tRNA. Now the methonyl tRNA is bound with initiation factor 2 and GTP molecule, and this particular complex, the complex of GTP, initiation factor, and methonyl tRNA, is called as ternary complex. And this complex will now go and bind to the P side. It will go and bind to the P side. It's not binding to the P side, but it will go and attach on the P side. And simultaneously the IF5, the initiation factor 5, is also coming and binding to the complex. Once the binding has been complete, then see, we are categorizing this 40S ribosome unit in terms of the Sydberg unit or the sedimentation rate. So when all these proteins, which are initiation factor and the tRNA, are getting attached to it, then of course its sedimentation rate will increase. Now the 40S sedimentation is increased to 43S, and this is called as 43S pre-initiation complex. When somebody asks you what is 43S, then you should be able to understand that 43 initiation complex is nothing, but eventually it's what? It's simply the 40S ribosomal unit along with all the initiation factors like 1, 1A, 3, 5, and methonyl tRNA. Then on the other hand, what is happening now, there is a requirement of mRNA. Until and unless the mRNA will be coming, there will be no translation, right? Then see, this mRNA is in linear form. This mRNA is currently in linear form. So we have a 5 prime cap, we have a poly A tail. To this poly A tail, there is binding of multiple proteins called as poly A binding protein, PABP. Then at the 5 prime structure, we have a secondary 5 prime and we have a secondary structure, and because of the complementary in these two regions, there is a looping, and this short segment will serve as double strand RNA. Now the initiation factor 4A is required to remove this double strand RNA structure because that's in helicase. Now initiation factor 4 complex will come along with 3. What 3? The EGA. The EGA, A is the helicase, and AG are the proteins. The E will go and bind to the 5 prime gap. G will go and bring this poly AB binding protein close to E, so that a loop is formed, as we had discussed in the earlier slides. And also there is a 4B, which and this binding is mediated at the expenditure of a ATP molecule. So what has happened now, the 4 initiation factor 4 complex has bound and resulted in a loop forming structure. Now this loop will be going and attaching onto the ribosome. This loop will be going and attaching on the ribosome. So the next step is attachment to the mRNA. The ribosome complex or the pre-initiation complex goes and binds to this particular mRNA. Now once it has bound, it scans the mRNA, it will scan the mRNA, and it will reach to the starting codon, Now it will align itself in such a manner that the P site reaches to the starting codon. The P site should reach to the starting codon. So when we are talking about the steps, let us see. The P site has now reached to this particular starting codon. Now before discussing ahead, let me show you one thing that majority of the time by default we skip. What is that? Once it has start scanning, that means the complete pre-initiation complex is moving towards the right side, moving towards the 5 prime to 3 prime direction. But what you observe, the 4E initiation factor is always bound to the 5 prime gap. The 4E factor is always bound to the 5 prime gap, and the remaining initiation complex is moving to the right, thereby shortening the loop. Fine, the loop is getting shortened. Then once it is aligned to the P side, once the ribosome has aligned the start codon onto the P side, the attachment of this formyl methonyl tRNA, which was not perfectly bound to P side, now binds to the AUG codon, which is recognized by the anticodon loop of this methonyl tRNA, and forms the hydrogen bonds. Once the bond is formed, there is a binding is initiated, there is an expenditure of the GTP molecule, which is attached to IF2. Now this complete unit where the mRNA has bound to the pre-initiation complex, the mRNA along with the initiation factor 4A family has bound to this particular pre-initiation complex, raises the sedimentation rate, or raises the Sydberg unit from 43 to 48S, and this 48S is called as your initiation complex. This is called as 48S initiation complex. Till now we have not reached to a state of ATS. We have not reached to the stage of ATS. But where we have reached? We have reached to a stage of 48S. 48S. Now once this mRNA has aligned itself at the starting codon, then next is what? Next is we have to ask the large ribosomal subunit that you should come and seal this particular complex to form an active ribosome which will carry out the protein synthesis. Then this work is performed by initiation factor 5. This initiation factor 5 will remove all the initiation factors, and then simultaneously there is another initiation factor called as 5B, which is bound by GTP. The 5B, which is bound with GTP. This 5B is responsible for bringing 60S ribosomal subunit, the 60S ribosomal subunit to the side, and then a properly aligned ribosome will be formed. What is happening? The E site present in the larger subunit will be binding to the same E site of the small subunit, P to P and A to A site. And if you see this pictorial representation clearly, carefully, then you will observe that all the initiation factors have been released, and thereby this loop which was formed is now there is no loop now. It's actually a linear strand. Why? Because 4G, which was responsible for forming the loop, is now dissociated. Once the 4G and 4A are dissociated, then there is no point of forming the loop. Now it's a completely linear mRNA. That's why I said that there is a transient state of the loop. There's a transient state loop. And then second point that is very much noteworthy, that all the initiation factors have been released, but only 1A was left bound to the A site. Only 1A was left bound to the A site, and this 1A serves as an adapter site for the binding of 5B GTP, which is actually required for correct orientation of the larger subunit onto the smaller subunit. Once the binding of the larger subunit or the alignment of the larger subunit has happened correctly on the smaller subunit, this GTP is hydrolyzed, thereby removing both the initiation factors from the complex, and then your complex is reached to a state of ATS, and this is called as ATS translation initiation complex, which contains the ribosomes, that is ATS, and methionine tRNA attached to P site. And this particular formation marks the end of your translation initiation stage. Next is what? Next is just elongation. Now the second codon codes for whichever amino acid, that amino acid tRNA will be coming and binding to the A site. Then there will be a peptide bond like we had discussed in the prokaryote. And similarly, as was in the case of prokaryote, the chain will continue to grow, and that process is called as elongation. So before going to elongation, if we summarize both the slides what we had discussed in just few minutes ago. So from first step to last step, we have discussed clearly what is mRNA activation, what is 43S initiation complex, what is the role of each and every initiation factor, and then what we have reached after initiation, right? So the step which is in which was a binding of initiation factor 4 to the mRNA, that is called as activation of mRNA. And the last part where we have reached is ATS initiation complex. So let us go ahead after having a clear-cut understanding of initiation complex and all the initiation factors. Now the ribosome is ready to elongate. And what is that stage? That is elongation. In prokaryotes, we discussed about 3 elongation factors, and similarly we have 3 elongation factors in eukaryotes also. And you'll be very curious to know that these 3 elongation factors that are present in eukaryotes performs the same functions as was in the case of prokaryotes. We read clearly that in prokaryotes we read about EFTU, TS and G, and similarly we have elongation factor 1 alpha, 1 beta gamma and EF2 to carry out the elongation in these eukaryotes. So what are these? These are actually the analogs. These are analogs to what? To the bacterial or the prokaryotic elongation factors. Now, see, this is how the elongation factor is going about. Now, what is happening? We had reached to this state in the previous slides where the initiation was over. Now, the next amino acid is ready to come. The next amino acid tRNA, I have denoted the first with the 1 and the next will be 2. So the 2 is bound to its tRNA. We are calling it as amino acid tRNA. It can be arginyl depending upon the coding. It can be glycinyl depending upon the code which is present next to the starting codon. So this incoming amino acid tRNA is bound with the first elongation factor called as 1 alpha. which is a GTP bound protein, and this elongation factor job is to deliver this particular amino acid tRNA to the A site. Once it reaches the A site, the GTP is hydrolyzed and the delivery occurs. The delivery of the amino acid tRNA onto the A site, and the elongation factor 1 alpha is released now. So if there are 20 amino acids and we have, suppose, let's say we have 10 amino acid long polypeptide. I am talking about a oligopeptide. We are not talking about very large chain. Let us assume that we have 10 amino acids that needs to join, then needs to form a polypeptide, or to form a peptide chain. Then if in every case this elongation factor 1 alpha is hydrolyzing the GTP and converting into GTP, then I need that many EF1 alpha GTP molecules, elongation factors in my cytoplasm, but it's not the case, because that will be wastage of the energy. Why you need multiple copies of elongation factor 1A? What you can do, you can remove the GTP and convert this released elongation factor 1 alpha back to its activated form, that is bound with GTP. And that exactly is the job of the next elongation factor, that was EF1 beta gamma, EF1 beta gamma. What it does, the EF1 beta gamma will come and bind to this particular 1 alpha, thereby releasing the GDP, and then it will transfer the GTP molecule to 1 alpha and will detach from the complex, thereby giving rise to a fresh elongation factor 1 alpha GTP, and then that particular molecule can go and bring the third amino acid, the fourth amino acid, and this is how the cycle continues. After this, let us focus what is happening on the ribosome. Now, the second amino acid tRNA is attached to the A side. Now, what is the requirement? Now, there is a requirement that there should be a formation of peptide bond between the one and the second amino acid. Who will form the peptide bond? Is it the spontaneous process? No. The peptide bond in case of eukaryote is catalyzed by 28S ribosomal RNA, the 28S rRNA which is present in the larger subunit. I told you in the RNA, when we were discussing the RNA types and structure, that 28S RNA is a kind of a ribozyme which has enzymatic function, and that is the function that it forms the peptide bond between the incoming and the pre-existing polypeptide chain. Then it will catalyze the formation of a peptide bond. Now these two, 1 and 2, are attached. Then what is the next step? The next step is, once they are attached, there will be a cleavage of what? There will be cleavage of the ester bond that was formed between the amino acid and the tRNA, which is present on a P site. Always remember. See, again focus on this particular ribosomal part. We have two amino acids which have formed a peptide bond. We have two tRNA molecules. Now there is a requirement to create a vacant space for the third incoming amino acid. Now what will happen? There will be a cleavage of the bond which is associating the amino acid with tRNA. There will be a cleavage. Fine. And that is the cleavage of ester bond. So the cleavage has happened, but this amino acid, the one amino acid, is attached to the second amino acid, so it will not go away, and this is how the chain is forming. Once the cleavage has happened, then another elongation factor, that is EF2, which is bound to GTP, will come. The elongation factor 2 GTP will come, and the GTP will be hydrolyzed to release the energy. And this EF2 is what? EF2 is nothing but an enzyme called as translocase. What it will do? The translocase causes the translocation. What translocation? Now the ribosome will shift towards right side. Now suppose the mRNA is static and ribosome shifts, and ultimately what happens? By shifting, the E site aligns itself to the cleaved tRNA, the P site is aligning itself to the bound tRNA, the dipeptidal tRNA, and the A site shifts to the right side to create a vacant space for the incoming third amino acid. And this is how the cycle continues. So you see, that is the elongation process, and that is the role of the elongation factors. Then once the cycle continues, a polypeptide chain keeps forming and the translocase keeps performing. So you see, like in, as was in the case of prokaryotes, in eukaryotes also, for adding addition of one amino acid, there is an expenditure of two GTP molecules. There is an expenditure of two GTP molecules, and that is how the elongation is brought about. That is how the complete elongation is brought about, and that what we have discussed is narrated here. What we have discussed is completely narrated here. Then what is the last part? Last part is termination. Now you have to terminate, and termination happens when the ribosome encounters a stop codon. What is a stop codon? UAA, UAG, UGA. These three are universal stop codon, whether it is prokaryote or whether it is eukaryote. Then there are factors in eukaryotes also that are responsible for termination of translation. In prokaryotes, we had RF1, RF2, RF3, releasing factor 1, 2, and 3. And here we have eukaryotic releasing factor, ERF. The ERF1 and ERF3, these two are very crucial for termination as well as the dissociation of the complete complex. So once the ribosome has encountered a stop codon at A site, what is the next thing? The complex of ERF1 and ERF3 bound with GTP will come and bound on the A site. These two releasing factors will come and bound to the A site. Then they will cause the cleavage of the ester bond, which is between the amino acid and the tRNA. Now that amino acid is eventually is a growing polypeptide chain, the complete polypeptide chain, and the tRNA which is present on the P side, it will cause the cleavage, thereby releasing the polypeptide chain. And this cleavage is again at the expense of this GTP molecule, thereby the energy is released and the phosphate molecule is removed. Also from the E side, from the exit side, the tRNA is released. So now what is happening? This post termination complex is made up of what? The termination has happened, then after termination still the ribosome is intact. It is intact with the tRNA as well as the releasing factors. Now what should happen? This tRNA should dissociate, but it never dissociates randomly. It is initiated by protein called as ABCE1. This ABCE1 proteins comes and binds to the complex and causes the dissociation of all the releasing factors, the tRNA, and both the ribosomal units. So thereby the complete termination complex is now released, is now separated. Two once both these units, the large and the small subunit, dissociates, then immediately the initiation factors, the first initiation factors that were there, the 1A and 3, we had studied these initiation factors on the very first step. They will come and bind to E site, A site respectively, and the 3 will bind to ribosome. And what is happening? When I told you before starting the initiation that both the ribosome units are never attached in cytoplasm, they are dissociated. They are in dissociated state. And this dissociated state is maintained by what? Is maintained by 1A and 3 initiation factors, because they are bound to small ribosomal subunit, and there is no space for premature binding of this particular ribosome until and unless these initiation vectors are released during translation. Then and then only this particular 60S ribosome unit will come and bind to the small ribosomal unit. And this is how the complete translation process is terminated. This is how the complete translation process is brought about and terminated. Now what has happened? The protein chain is now released from the ribosome and has reached to the cytoplasm. Then this protein will start folding and will undergo multiple modifications. What are those modifications? See, modifications in terms of like acetylation. In almost 50% of the eukaryotic proteins, you will see that the amino terminal of the polypeptide chain is always acetylated. The acetyl group is attached after translation, resulting in the modification of the protein. When the final functional protein after the three-dimensional conformation has taken place, then you will see that in majority of the cases the N terminal is acetylated, whereas whereas in prokaryotes it was formulated. Then see, every protein once formed in cytoplasm, suppose it is destined towards uh, endoplasmic reticulum, or it is destined to go back to the nucleus, then there is a signal sequence, there is a signal peptide which is present as a leader sequence. Then once it has reached to nucleus with the help of nuclear localization signal which is present as a signal sequence, then that signal sequence is cleaved off to give rise to a chain which is actually a protein which will fold properly to give rise to a functional protein. When the signal sequence has done its job, the signal sequences are 15 to 30 residues of the amino terminal, and in majority of the cases these signal sequences are removed by specific peptidases. Then there are certain cases where there is phosphorylation, because in majority of the pathway enzymes are getting activated by phosphorylation. The signaling pathways are conducted because of the activated and inactivated state. The activated state is phosphorylated state, whereas the inactivated state is non-phosphorylated state, whether it is p38 kinase, whether it is JNK, whether it is ERK, the phosphate form is always active. Even in the case of glycolysis, when you're talking about phosphofructokinase, the fructokinase is converting into phosphofructokinase. Now again, an enzyme is getting converted into active form by attaching a phosphate group, and that is called as phosphorylation. Now where this phosphate group will attach? Of course, by phosphodiester bond. And where this particular bond is forming? Of course, with the anhydride bond. Now where this particular ester bond will be forming? It will be forming with the OH group, the OH group of the amino acid. What amino acid? The serine, threonine, and tyrosine. These three amino acids will go for phosphorylation. Like example of milk protein, the casein molecule, it contains phosphocyrene groups which binds to the calcium sulfate. And similarly there are multiple examples that what I have narrated. Apart from this phosphorylation, what happens? The N-linked and O-linked glycosylation, the addition of carboxyl moiety, like in case of glutamate, when the carboxyl group is attaching, the carboxy group is attaching, actually CO group is attached to form gamma carboxyglutamate. You can see here at the side chain there is an addition of a carboxy group on the carbon. And what is happening? This is observed in prothrombin, the blood clotting protein. After this particular prothrombin protein has been shown to contain multiple carboxyglutamate, which is responsible, and this, of course, this is responsible for blood clotting, or that is required for vitamin K, or this carboxyl group is actually what it does. This carboxyl group goes and binds to the calcium ion, which is required for initiating the clotting mechanism, and that is why it is very much crucial for prothrombin. Likewise, the methylation and the addition of methyl groups, one addition of a single methyl or addition of double methyl or trimethyl group happens frequently in many cases, like methyl lysine or dimethyl lysine or trimethyl lysine or methyl glutamate. These have been shown to be present in multiple cases, like in muscle cells, the methyl lysine has been shown to be present, or dimethyl lysine, the attachment of methyl group to the lysine residue has been shown to present in muscle cells, or in cases of cytochrome C. And in case of glutamate, if the methyl glutamate is present in calmodulin protein, which is a calcium binding protein, which is again a very important protein in mediating the calcium based signaling pathways. So in nutshell, what I'm trying to say here is that the protein, after being formed and then folded properly to give rise to conformation state, in between that it requires certain post translational modifications, that what we are discussing right now, and these modifications play a very crucial role when we are talking about in terms of properly functional protein. So when I talked about the addition of a carbohydrate moiety on the particular probably thread chain, that can happen at either n linked, or that can happen via O linked. When I'm talking about the n linked, where I'm talking about the attachment of carbohydrate group to the asparagine residue, whereas when I'm talking about the addition of carbohydrate group to serine or threonine, we are talking about O linked glycosylation. So you see this n linked and O linked glycosylation leading to the formation of glycoproteins leading to the formation of glycoproteins are very crucial for what? For mucosal membranes. For mucosal membranes. And there are multiple glycoproteins that are present as receptors on the membrane which carry out essential functions, whether it is immune system or whether it is some other pathways. Then addition of isoprenyl group, like in the case of frenesyl pyrophosphate, the expression of RAS protein, which is a product of the RAS oncogene, a proto-oncogene, and many other proteins like G proteins or lamin proteins. What is happening? An additional additional isoprenyl group called as frenesyl group is attached is attached to these particular proteins by linkage. See this particular thiol group which is present, thiol moiety which is present, the cysteine residue contains the thiol group. This cysteine residue forms a bond, a thioether bond, to give rise to an attached phenesylated RAS protein. So you see, there are multiple post-translational modifications which are resulting in which are happening in eukaryotes. They never happen in prokaryotes. They are happening in eukaryotes, and that are very much essential. In terms of executing the desired function of that protein. Like if you talk about the immunoregulatory transcription factor NF-kappa B, it is always in the inhibited state. Once the P65 subunit is getting phosphorylated, then only it becomes activated, and then the dimer is formed between P65 and P50, and then it goes and binds to its response element, thereby causing the expression of pro-inflammatory cytokines. Or if you talk about the NRF2, the NRF2 is again a transcription factor, the immunoregulatory transcription factor which actually is responsible for detoxification, which is actually responsible for binding to the antioxidant response element in order to substantiate the antioxidant proteins, that's antioxidant machinery, whether it is glutathione, whether it is glutathione peroxidase, or whether it is glutathione transferases, or whether it is heme oxygenase 1 or NADPH oxidases. What happens? The NRF2 gets phosphorylated. The phospho-NRF2 then becomes activated and goes and binds to the antioxidant response elements. So you see, post-translation modifications are again very much essential in terms of carrying the proper functions of these proteins. And few other I have mentioned here, like addition of prosthetic groups, as in case of hemoglobin, or when we're talking about the Barton molecule, which is very much required for acetyl coenzyme carboxylases. Until and unless the Barton is available, it will not perform its function. Or the heme group, which is required for cytochrome C to perform its function. Then we have a proteolytic processing also. What is proteolytic processing? When the zymogens are converted into active enzymes, like trypsinogen after proteolytic cleavage is converted into trypsin, or proinsulin, which is secreted from pancreas, undergoes proteolytic cleavage to give rise to insulin. Or the viral proteins that are formed as a single polypeptide, and the viral protease cleaves those peptides to give rise to functional proteins which are responsible for viral replication or viral infection. Then another aspect is formation of disulfide bond, which is happening inside the cysteine between the cysteine residue. The disulfide bond is responsible for proper folding and the cross-linking of the protein to give rise to a three-dimensional conformation structure. So all these modifications are giving rise to a properly conformationally active properly folded protein molecule, which is actually the product of a translation. Now translation has given you a linear protein chain made up of amino acid, a polymer of amino acid. Now in order for that polypeptide chain to fold properly and give rise to conformation protein or an enzyme, these all modifications play a very crucial role. So this is all about, students, today with the translation in eukaryote part. This is again for your reference. I've given the links of further reading so that you can go ahead, read these articles and further increase your understanding of this particular topic. Now let us brush up with these MCQs what we have put forward, whether in terms of eukaryotes, the translation is initiated by what? The binding of ribosome. to what? The Pribnow box, the Huggins box, the 5' cap, or the poly A tail, or which one is the initiation complex in eukaryote, or how many initiation factors are we talking about in eukaryotes, whether we are talking about ribosome recycling, when we are talking about whether it occurs in prokaryotes or eukaryotes, whether they are always intact or whether they are loose and again forms intact ribosome, then which amino acid is responsible for formation of disulfide bond, or which amino acids are responsible for getting phosphorylated, or ef1a, where it is binding, one is responsible for binding to E, P, or A site, or when we are talking about another case, that when which actually RNA is acting as ribosome to form a disulfide bond, whether it is 28s, 5s, 18s, or 5.8s. So these MCQs will help you in brushing up what we have understood in this particular lecture. So with this, student, I thank you very much for your patience. I thank you very much for your keen listening. I hope the our projected outcome is now achieved, and you have a very clear understanding of how this particular translation in eukaryotes is happening. So with this, I will take your leave, and I'll be back with another lecture with in this particular series of molecular biology. Till then, please stay safe and wish you a very happy learning. Thank you so much.